Our next speaker is Zhaolu Liu from Imperial College London, and the title of his talk is Physics-Informed Deep Learning Approaches to Restore and Enhance Hyperspectral Images of Brain.
Yeah, thanks so much for the introduction. So I'm Zhaolu Liu, I'm from Imperial College London, and the thing I'm going to present here today is physics-informed deep learning approaches to restore and enhance hyperspectral images of brain. So hyperspectral imaging is vital in biomedical fields like brain imaging. It's able to distinguish tissues via spectral differences for diagnostics and surgical tools. And mass spectrometry imaging is one of the form of hyperspectral imaging technique, and is able to spatially map thousands of chemicals simultaneously. And the advantage of MSI, the mass spectrometry imaging, is that it has very high spectral resolution, but usually sacrifices speed for spatial resolution. So for this physical limitation, here we're proposing a computational solution, which is what we call high-res. So using high-res, we basically overcome all these challenges, and so we don't have to deal with the trade-offs anymore. And in particular, this is a deep learning-based method that is able to do image restoration and image super resolution. Right, so the key to the success of these deep learning models is you have to have good quality of training data. So what does that mean is, in this particular case, we need to model from the LR, which is the low resolution images, to generate the high resolution images. And in biomedical setting, this is pretty complicated, right? Because you generally don't image both the LR and the HR at the same time. So what we do is we synthetically generate the LR by treating the original images, ground truth images, at the HR, and we apply the bicubic sampling, downsampling with the consideration of detector noise to generate physically meaningful HR and LR pair to fit into the model. And so that's one key, right? So the other key to the success of these deep learning models is not only you need a very good quality of training data, but you actually need a lot of them to scale. And again, this is pretty simple, right, in terms of natural images, because you can simply just download loads of them from online resources, right? But this is very, very difficult, actually, in biomedicine, as you probably all know, because it's, first of all, it's probably time-consuming, it's usually very expensive. So the approach that we are leveraging here is we leverage transfer learning. So basically, we fine-tune an open source super resolution model that has been pre-trained on natural images. So we fine-tune that with a novel loss function called the Fourier ring correlation loss function that is basically calculating the resolution in the frequency domain. Right, so what is the main advantage of high-res? So what is able to do? So the main advantage of high-res framework, it is able to improve the imaging speed massively without any compromisation of the resolution. So we train our model using the HR, the HR synthetic generation process that we just talked about previously, and we apply it to the experimental LR. So apart from clearly enhancing the resolution of LR inputs, the high-res restored output images are also observed to have visibly improved SNR, so the signal-to-noise ratio, and very high quality when you compare it against the HR ground truth images. So to quantitatively illustrate this enhancement, we actually see that both the CRIS score, so this is an image quality metric, and also the resolution based on the FRC calculation, so both of those metrics improve. So we also, so at the start of this slide, I was saying, the main advantage is it increases the speed, right? So in terms of imaging speed, it took about 6 hours to experimentally acquire the HR images. But actually, if you just get the LR images, it would take around 30 minutes. And with additional of 3 minutes, you'll be able to get like very comparable results to the experimental HR using high-res. So we say, we improve the speed and also the image quality, but does the high-res restored images have retained the biomedical information of the original images? And the short answer is yes. So we apply high-res to a multiple sclerosis data set. So multiple sclerosis is a chronic autoimmune disease of the central nervous system, and it's the leading cause of lots of disabilities worldwide. So previously, we have shown that using these spinal cord samples from healthy and multiple sclerosis mice, we can see very distinct metabolic patterns. So this is unveiling a new mechanism for multiple sclerosis progression that may be targeted by a novel treatment. So what we do in this case is we apply high-res on the same multiple sclerosis data set that have been downsampled and noise corrupted. So from the ROT curve, you can see in this slide, we can see a sharp decrease of 50% in the ROT curve using the LR to do the disease progression, disease detection. And this is basically induced by the artificial downsampling. But this is nearly completely restored by, if you use the high-res on the LR images. And I just wanted to note that, this model has never seen the data set before, so it's not trained on the multiple sclerosis data, it's actually trained on a different data set. So we're not claiming that we have a universal model, but it's actually, we're trying to say that, the model actually can detect the useful biological information in the data set. So the restored images yield the same patterns with the same significant metabolites, and we can see little to no hallucination effect in the high-res restored images. And one more evidence we show is that the mean spectra between the high-res images and also the high-res restored images are basically identical. And this is sort of confirmed by a Spearman correlation coefficients to be very close to 1. So the actual coefficient is 0.998. So in addition, actually high-res is able to enhance the biomedical information. So it's actually useful for downstream analysis. So we use high-res on a Down syndrome data set. And Down syndrome is basically one of the most common genetic disorders worldwide, and is hallmarked by an additional chromosome 21. And recent studies consider Down syndrome as a metabolic disease, and this makes mass spectrometry imaging very useful, and it basically provides a unique tool for spatially study the brain metabolic profiles of the Down syndrome mice. So what we do is we apply high-res to the ground truth images, resulting in a fourfold upsampling in both dimensions, the X and Y dimensions. And to test their sensitivity to trisomy, we use regions from the cerebral cortices to train a classification model that are optimized via feature refinement method, and then used to predict the Down syndrome pixels in both the training images and the test images, independent test images in this case. So we see that from the ROT curve, the high-res model retains superior performance, so similar to the multiple sclerosis data set we have seen. So in this case, it has a AOC of 83%, whereas the ground truth model is basically as good as just a coin toss, which is roughly speaking 50%. So the high-res images make it really easy to tell the trisomic brains from the disomic ones with higher and more concentrated prediction scores. And even though the model was only trained on the cortex pixels, it will still picks up clear hotspots in areas like striatum, forebrains. These regions are known to be affected by the trisomy of 21. And very interestingly, we find that the high-res also revealed a social effect from the housing setup. So before actually imaging the healthy and Down syndrome mice, the healthy mice are living with the trisomic ones. It's basically they're co-caged in the system, and we find out that these disomic mice that are living with the trisomic mice are more likely to be misclassified by the model. So this is showing that very high pixels in the region of dorsal striatum, which is a region that's responsible for habit formation. So why is this powerful, right? So why is the high-res framework so powerful? So to interpret the strong restorability of the high-res, we further investigate the physical effect of the model on the imaging system. So some of you may know that the current mass spectrometry imaging lacks in spatial resolution. So therefore, it struggles to provide cellular level, detail that is essential for scientists and clinicians. So previously, we concluded in a different paper that state-of-the-art resolution images such as the ER-SAGAN that are able to do enhancement is unable to go beyond the instrumental limits. But using high-res, we actually see that a theoretical pixel resolution of 2.5 micron, which brings MSI images very close on par with the optical equipment, which has a pixel resolution of 1 micron. And by comparing it with the IMC, which is the single cell level optical image, we can see there is clearly some spatial overlaps. And this is for the first time that the mass spectrometry imaging is able to recover the single cell level detail. So just to summarize, the advantage of high-res is it's really fast, and it's artifact-free, and it's also able to enhance the downstream analysis and reveals new biology. And if you have any further questions, I also have a poster in the poster session today. And yeah, thank you so much.
Thank you so much for that. We are actually out of time for questions, but please submit your questions on the app or come up after the talk. So thank you very much. Let's thank our speaker again.